Hello, is anyone there? All right, sorry about being late. Come on, turn on. There we go. We will get started shortly. So what we're doing today is chapter 14, Principles of Disease. Are there any questions? There is no lab today. I will be on at 6:30 to 6:45 to answer questions. All right, if there's no questions, let's go ahead and get started. Chapter 14. So we had finished talking about this slide, classifying infectious diseases. Let's move on to the next terms, classifying infectious diseases. There are communicable diseases, and that is any disease. Did we talk about this? Let me see if I have another two here. Ah, yeah, we talked about that. Sorry, we're going to be finishing up the control of nosocomial infections, and we did talk about nosocomial infections, and you should use certain procedures for controlling them because you do not want nosocomial infections when you are working in a healthcare setting. You do not want the patients you are treating to come down with a nosocomial infection. So take all the steps you can to prevent their spread. Any questions about any of that? All right. A few terms we need to note about epidemiology. Epidemiology is the study of where and when diseases occur and how they are transmitted in populations. The CDC, that's the Center for Disease Control, collects all that information and analyzes it in the United States. And they gather that epidemiological information and publish it in the Morbidity and Mortality Weekly Report, which is so long that most people just abbreviate it the MMWR, and it's published by the Centers for Disease Control. There are notifiable diseases that physicians and other healthcare workers are required by law to report. And generally, you report them to the local county health authorities, and then they report them to the state health authorities, and then the state authorities send that directly to the Center for Disease Control. Occasionally, that process will be interrupted, like with COVID-19. I think that information is directly sent to the Centers for Disease Control. It's not, well, it's also sent to the other places, but the doctors, I think, are supposed to send that directly to the Center for Disease Control. And that's just to get the information more quickly. All right, some other terms of epidemiology. Morbidity is the incidence of a specific notifiable disease. The morbidity rate is the number of people affected in relation to the population in a given time period. The mortality is the deaths that occur from a notifiable disease. The mortality rate is the number of deaths from a disease in relationship to the population in a given time. And does anybody know the mortality rate for COVID-19? I don't know what that would be for the number of deaths in relationship to the total population in a given period of time. But we will talk about that. It's estimated that the mortality rate, at least of elderly or older individuals, ranges from something like 1% to, especially the very elderly, to up to 10%. And they have an estimate for the number of people in the United States that die from COVID-19, assuming they get infected. And I'm afraid I don't remember what that number is. It's something like 0.4% to something like 2.3%. And we'll discuss that when we're talking about viruses in our next lesson. Did I close that down? I sure did. Let me see what our next lesson is. I think it's viruses. Well, is that going to open? I clicked on it. Yes. Let's get started with viruses. And I don't have that here. Let me get my thumb drive on. Oop, that's the wrong one. Chapter 13. All right, like always, I show the major goals, and I think I have to save that. Major goals of the slide and the rough outline. So know the terms: a capsid. What's a capsid on a virus? An envelope, spike, bacteriophage, and the different life cycles of a virus: a lytic life cycle, a lysogenic life cycle, prophage, provirus, and what are latent and persistent infections. Any question about any of that? Let me close that down. And then plug in my earphones. All right, no questions. Let's move on. Oops. Also understand the characteristics, the structure, and the classification of viruses. Understand about viral multiplication. We'll talk about the different viral life cycles: lytic, lysogenic, latent, and persistent infections. Understand about prions. These are not viruses. And about viroids. Any question about that? All right. The only goal a virus has is to replicate. And how do viruses do that? Well, they invade a host cell, hijack the host cell, and then force that cell to make viral copies, which then escape from the host cell. And let's see if I can open this. I'm not hearing sound. So a phage is a term for a bacterial virus, and we'll call them bacteriophages. All right, any questions about any of that? Okay, so some general characteristics of viruses. Viruses are inert when they're not within a host cell, meaning they're not metabolically active when they're not inside a host cell. They require a living host cell to multiply. Viruses are obligate intracellular parasites. They cannot multiply, and really they are not metabolically active unless they can infect a host cell. They multiply within living cells by using the host cell's enzymes, nucleic acids, amino acids, and other molecules. Viruses have no ATP generating mechanism. They have to rely on ATP from their host cell. They contain either DNA or RNA for their genome, but viruses do not contain both. They have either DNA or RNA. And the DNA or RNA is the central molecule, the genome of the virus, and that genome is always surrounded by a protein coat. Some viruses, this virus has the edge cut off so you can look inside of it. Some viruses, besides having a protein coat, have an outer lipid envelope surrounding the protein coat. Not all viruses have an envelope. The host range of a virus is the cells that a virus can infect. This is determined by specific host attachment sites, meaning the cell walls, the flagella, and other cellular factors, which are the receptors that the virus binds to. It's also dependent on the attachment factors of the virus that bind to the receptors. Most viruses only infect the cells of one species, one host species. That isn't true for all viruses. Like we've discussed, the flu can infect humans and pigs and horses and birds. But the flu is an unusual virus in that it can infect more than one species. And the flu can occasionally, not often, but can occasionally infect dogs and cats as well. Most viruses only infect, like I said, the cells of one species. Like HIV, essentially only infects humans. It can infect one of the other chimpanzees' cells, but it does not give the chimpanzee AIDS. And polio is another virus that, as far as I know, only infects human cells. And those are much more common for viruses, where they only infect essentially the cells of one species. When we look at the size of viruses, we find, let me see if I can blow this up, we find that they are much smaller than a eukaryote cell, where part of one is shown here, and much smaller than a bacterium. And E. coli is a little bit larger than your average bacterium. The Ebola virus, lengthwise, is about the biggest virus, but if you talk about the volume of a virus, megavirus is one of the biggest viruses. And most viruses are smaller than these two viruses. The smallest virus that you have probably heard of is the polio virus. There is a smaller virus. Is that M52 or M52 or MS2? I can't read it even though I've got this blown up. It's MS2. I'm sure no one's ever heard of MS2. Like I said, poliovirus is probably the smallest virus that you are familiar with. Let's move on to the viral structure. The viral structure contains all of the elements you can find within a virus. A virion is a single, mature, complete infectious virus particle. So usually when we're talking about a virus, we are referring to a virion. But a virion is usually used when the viruses have replicated in a cell and then are released. Inside a virus, you can find a nucleic acid. And this is the genome of the virus. You can find either DNA or RNA, but you do not find both molecules in a virus. You either find one or the other. Any questions about that? Am I sharing my screen? I'm not sharing my screen. Somebody should have mentioned that. The nucleic acid can be DNA or RNA. Furthermore, the nucleic acid can be double-stranded or single-stranded. So we have viruses which are double-stranded DNA viruses and viruses which are single-stranded DNA viruses. Both of these are seen in a eukaryotic or a bacterial cell. You have single-stranded DNA. Whenever the DNA is replicating or being transcribed. You also have, for the RNA viruses, double-stranded RNA viral genomes. And this is unheard of inside a cell. We do not have double-stranded RNA. And then we have single-stranded RNA viral genomes. And when we're talking about single-stranded RNA viral genomes, you have to realize that viruses can be positive single-stranded RNA or negative single-stranded RNA. If the viral genome is positive single-stranded RNA, that means the viral genome can be read by the ribosomes. If it is a negative single-strand RNA viral genome, The genome of this virus cannot be read by the ribosomes. The negative single-strand RNA genome is the complementary to the positive single-strand RNA genome. Viruses can be linear, where the genome starts where it ends, or, excuse me, did I say that right? If it's circular, the viral genome starts where it ends. If it is a linear genome, then the starting position is different than the ending position. And I think this virus here is a linear because it looks like it starts there and ends here. Some viruses, like influenza, have their genome in several segments, but most viruses do not have their genome in several segments. And if you're wondering, humans have their genome in several segments, and that's why we have 46 chromosomes. When we're talking about viral structure, as I mentioned, all viruses have, besides their genome, a protein coat surrounding the genome. That protein coat has a name, and it is called the capsid. The capsid is composed of capsomeres. Let me blow this up. So when we're looking at this virus here, you can see that the protein coat called the capsid is composed of many units of capsomeres. And here's one here, there's one there, there's one there. And this virus, the capsid is composed of one capsomere. Some viruses have different proteins making up the capsid, so they'd have different capsomeres, but this virus only has one. And then I stated that some viruses, in addition to having a protein coat, the capsid, they have an envelope, and that surrounds the protein coat. If it has no envelope, the virus is called a naked virus. So this virus is a naked virus, and this virus shown here is an enveloped virus. The envelope is a lipid layer. It also has proteins and carbohydrates in it, and it's mostly a bilayer of phospholipids, and that should be familiar to you, and we'll talk about why the envelope is similar to a membrane of a cell. If the virus has an envelope, it may be covered with a glycoprotein spikes. So these, I don't know, pink or reddish things sticking out of the virus, those are the spikes. And spikes are only seen on enveloped viruses. The spikes are antigens, which are usually the outermost molecule on a virus, and antigens are something that the host immune system recognizes as foreign and then responds to. So the spikes of viruses usually serve as an antigen. And of course, that's not the purpose for the spikes on a virus. The spikes usually serve as an attachment point for the virus to bind to their host cell. And it is always the external-most molecules of a virus that serve as the attachment point for the virus to bind to their host cell. Now, on a naked virus, the capsid is the most external molecule of the virus. So the capsid is what binds to its host cell. Any question about any of that? If not, let's move on about viral structure. We can talk about the general morphology, that is the shape that viruses may be classified on. So we can classify viruses on their shape, and our author classifies viruses on their shape into 3 classification systems. There's a helical virus, shown here, and I don't see the helix in there, so we'll just talk about that one. There are polyhedrons, and realize this is cut away, and there's an actual electron microscopic image of a polyhedron virus. And then there are complex viruses, and they can take on many different shapes, like that, or like that, or like this. And of course, there are enveloped viruses, and an enveloped virus does not have to be a complex virus. We'll just leave it like that. This virus here is bacteriophage T4, and we'll talk about it because it is the most studied of all viruses. It does look like the lunar lander, if you remember from the Apollo missions. It looks like the lunar lander, the part of the Apollo mission that landed on the moon. T4 is in the class of viruses that we call the T-even virus, including T2, T4, T6. They all have this shape where they have a protein coat, which is very different in different sections. This is the head of the capsid, and inside the head we have the DNA molecule or the genome, and then this is the sheath, which is the protein coat or the capsid, and it's a different molecule than the head. And then there's the base plate, and then the tail fibers. Let me just state that the tail fibers are what T4, as well as the T-even bacteriophages, bind to their host cell. So this part right here, and there, and there, and there, are where this virus binds to their host cell. Any question about that? The reason why T4 is the most studied virus is simply because this virus infects E. coli, and people wanting to study viruses wanted a virus that infected E. coli, a cell that is very common in the microbiology labs. And so T4 has been studied more than any other virus, and that includes more than any other human virus. Now let me state that a bacteriophage is a virus that infects bacteria cells. And so bacteriophage will be the term we're using to discuss a virus that infects a bacteria cell. Viruses, we haven't talked about this, but they tend to infect one kingdom. In fact, I can't say 10. As far as I know, all viruses infect cells in one kingdom. And you can actually classify the virus by what kingdom it infects. And so there are animal viruses, and plant viruses, and fungi viruses, and bacterial viruses that we'll call bacteriophages. And then there's other viruses, like viruses that infect archaea cells. Any question about any of that? All right. Viral infection and the host immune response. Infection with a virus of an animal cell stimulates the host to produce antibodies. Antibodies recognize and bind to viral surface proteins, meaning they're binding to the antigens of the virus. Some viral proteins can change or mutate frequently, meaning that different strains of the virus can emerge and therefore can infect the same individual more than once. A best known example of this is the influenza virus. And that is why you can have the flu many times over many years. Any question about that? It is also why when you get a vaccine for the flu, it's only good for 3 years because the virus is changing over time, and after 3 years the virus will have changed enough that the flu vaccine is no longer protective. Now that's a simplification. Let me, I don't know if I can get that to work there. So let me try and, oh, that's the wrong picture. Gotta edit that. Explain what I'm talking about. And that is, with time, the vaccine decreases. And here's our graph. And once the vaccine drops below a certain threshold, we say that it is no longer protective. So with time, a vaccine decreases in its effectiveness. And after 3 years, it drops below a threshold where we say the vaccine is no longer effective. In reality, the vaccine has a little bit of activity, but it's below the threshold. Any question about any of that? So viruses are grouped into families, and you will be pleased to know when we're talking about viral taxonomy that we don't group viruses other than the kingdom they infect. We don't group them into different categories like, let's see, subphylum and phylum, and is it class and order? Some of them have one of them. Is it an order? King Peter's son came over. Yeah, some of them have orders. But generally we group viruses into families, and that family is based on the type of nucleic acid the virus has, the replication strategy, their morphology. All family names of viruses end in viridae, and then we group them, the family, into a genus. The genus viral name ends in virus, like herpes, genus, virus. The viral species is then the next viral classification, and a group of viruses, a species is a group of viruses sharing the same genetic information and the ecological niche, meaning they infect the same host cells. You'll be pleased to know that viruses do not have a Latin scientific name. We use common English names to name all viruses, like the polio virus and herpes simplex 1. These are common English names, and apparently the first people studying viruses were all English speaking, and that's why we use English names for viruses. Now, with viruses, it's important to realize that we also classify them into subspecies, and the subspecies are designated by a number. Like you can have herpes simplex type 1 or herpes simplex type 2. And the subspecies are very different, as you probably know from herpes simplex type 1 and type 2, they're very different. One is a disease causing the cold sores, and the other is a disease causing genital lesions. But let me state how different a subspecies is. One subspecies of the polio species of virus infects people and causes the disease polio. The other subspecies of polio is what we use for the live polio vaccine to prevent people from getting polio. So this is showing you that one species causes the disease polio, and the other species is used as the vaccine to prevent people from getting the disease polio, showing you how important it is to recognize that the subspecies of viruses are very different from each other, even though they're the same species. All right, a little bit more about viral taxonomy. An example, the family name, herpes viridae, the genus, simplex virus. The common name would be, instead of simplex virus, the human herpes virus, or herpes simplex. The subspecies would be human herpes virus type 1, or more commonly called herpes simplex type 1. Now, let me state that the human herpes virus is the new scientific name for this group of viruses, but that term has not been widely adapted, that people are continuing to call the viruses herpes simplex type 1. So they're continuing to use the old name, and that is true for clinicians as well as just most people in general. All right, let's move on and talk about growing bacteriophages in the lab. I've actually done this. Viruses can be grown in the lab. They have to be grown in a living host cell. And bacteriophages can form a plaque on a lawn of bacteria. So, how do you say this? You plate the agar plate with bacteria so that the bacteria are growing everywhere. That's why we call it a lawn of bacteria. The bacteria growing everywhere, so it looks like a nice lawn, like in your front yard. The grass is everywhere. And then you, before the bacteria grow up, once you plated the bacteria on the plate, you then put a solution that contains the bacteriophages. And wherever a bacteriophage infects the host cell, it will infect that cell, replicate, lyse the cell, releasing the virus. And where do those viruses go? To nearby neighboring bacteria cells. And what do those do? They infect those cells, replicate, lyse those cells, and then go on to more neighboring cells so that we can see the number of bacteriophages in solution by counting the number of plaques, because each plaque is where the bacteria cells have been lysed, and that's why it's clear. That's the color of the agar. The whitish color here is from, or maybe I'll go there, is from the bacteria growing. And wherever there's a plaque, there was an initial bacteriophage that went on to replicate and went on to form the plaque, where all the bacteria cells are lysed. And so you can count how many bacteriophages were in this solution here. And this is a large number, I'd say almost 200 or more. Oh yeah, why is this colony bigger than this colony? That colony looks like that was more than one colony that merged to make that big colony. But this colony probably is larger than this colony because the bacteriophage initially infected a bacteria, so the virus started replicating immediately, whereas this virus did not infect a bacteria immediately. It had to wait until a bacteria was growing, and then a bacteria came to the bacteriophage, and then the bacteriophage began replicating. All right, let's talk a little bit about viral multiplication. The viral genome only contains a few genes, so viral genomes are very small compared to a cell. They only have a few genes, and these include the capsid proteins that are needed by the virus, as well as perhaps a few enzymes that the virus needs. Everything else the virus needs, it gets from hijacking its host cell, and then it uses the molecules of the host cell. So following an infection, the virus takes over the host cell's metabolic machinery and then uses the host cell's metabolic machinery to replicate the virus. And the virus will use the host ribosomes. Viruses have no ribosomes. They will use the host tRNA, the host amino acids, the host ATP, and some of the host enzymes. Okay? The basic mechanism of viral multiplication is similar in all viruses. The bacteriophages, and some people, like some of the movies I have, will call them phages. We mean bacteriophages, viruses that can infect a bacteria cell, are viruses that infect bacteria. Bacteria viruses use two mechanisms to replicate. They use the lytic life cycle, where the host cell is infected and then lysed, and the host cell dies when it's lysed. And the bacteriophages could also use the lysogenic life cycle, and it depends on the species what life cycle they're using. If they use the lysogenic life cycle, the bacteriophage infects the host cell, but the host cell remains alive, living in symbiosis with the bacteriophage genome. And we'll talk more about that probably on our next lesson. I don't think I'll get to that today, because we have to talk about the lytic life cycle. Although we might. Let's see how we do. So what do new bacteriophages need? What do they need? What components do they have to make in order to replicate the virus? Well, if they're a DNA virus, and it depends obviously on what type of a genome the virus has, and we'll take a look at that a little bit later, but here we're just talking in general. What they need, the DNA has to be replicated so that there can be multiple copies of the viral genome. That viral genome has to be transcribed into viral messenger RNA, and that viral messenger RNA has to be translated into viral proteins, the most important of which are the capsid proteins of the virus. And then the capsid proteins have to get together with the viral genome to make the new progeny virions. So these are what we need to replicate the viral, not genome, to replicate the viruses, okay, to make new virions. So let's talk about the lytic life cycle. It occurs in 5 stages. Oh, that's weird. Oh, I know what the problem is. All right, let me talk about the the 5 stages of the lytic virus, and I don't have the movie on my computer, so I've got to put in my thumb drive. So the first stage is attachment. Attachment, the virus attaches to the host cell, and the phage will attach using attachment cycles, attachment molecules, and those will bind to the receptors, and then the bacteriophage is attached. And then that followed by penetration, where the phage lysosome opens up the cell wall of the bacteria cell so that the virus can get in. Is that the one I want? Don't remember. And the tail sheath of the virus contracts to force the genome of the virus into the host cell. Once the genome of the virus gets inside the host cell, begins the phage of biosynthesis, and that will be the production of new viruses. And replicating the viral genome and the protein coat proteins. That's followed by maturation, where you have assembly of the phage particles, where the protein coat and the viral genome get together. And that's followed by release, where the last thing a lytic bacteriophage do is it secretes the enzyme lysozyme, which breaks down the cell wall. That will lyse the cell, and then the host cell dies, releasing the new virions. Any question about any of that? If not, let's watch the lytic lysogenic life cycle.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall and the nucleic acid from this capsule is injected through the hollow tail into the cell.
Let me back that up just a little bit and show you right there. The sheath contracts, and that puts pressure on the head so that the viral genome is injected into the cell.
And the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes.
All right, we'll continue this with the lysogenic life cycle. So I'll leave this up and pull it back, about right there. And we'll see if we have time to show you that in this lesson. So in the lytic life cycle, just going over it one more time, the virus attaches to the host cell, opens up a little section of the cell wall, the sheath contracts, it injects the viral genome into the cell, the viral genome is replicated and transcribed. The messenger RNA of the virus is then translated into viral proteins. The protein coat of the virus gets made and assembled. Looks like I'm missing something. Biosynthesis. Maturation occurs where the protein coat and the viral genome get together, and then release where the virus releases a lysozyme, an enzyme that destroys the cell wall that will lyse the cell, releasing the viruses. Two terms you should know about with the lytic life cycle is the burst time. That's the time from the phage attachment, the initial phage attachment, to the time that the new virions are released, the burst time. The burst size is the number of newly released, newly synthesized virions. That's usually between 50 and 200. You don't need to know the number, but the burst size depends, as well as the burst time, depends on the viral species. And different species will have different burst time and burst sizes. All right, we probably have time to talk about the lysogenic life cycle. The lysogenic life cycle is the second life cycle that bacteriophages may engage in upon attachment of a bacteriophage and penetration of the host genome. Two things can happen with a lysogenic virus. The lytic life cycle can happen where the virus then replicates, lyses the cell, and then the virus is released. So a lysogenic virus can engage in the lytic life cycle, or it can engage in the lysogenic life cycle. In the lysogenic life cycle, the phages remain inactive, sort of latent, and the bacterial host cell is termed a lysogenic cell because it has a lysogenic virus within it. And the host cell, remains alive in a state of symbiosis with the lysogenic virus. So initially, when a lysogenic life cycle virus infects a host cell, it injects its genome into the virus, or into the host cell, and it can enter the lytic life cycle, where the genome of the virus remains separate from the host chromosome, and then the genome is replicated, the viral proteins are replicated, the viruses are made, the host cell is lysed, and the newly replicated viruses are released. But upon entering the host cell, a lysogenic life cycle can also, it either decides immediately to enter the lytic life cycle or enter the lysogenic life cycle. If it enters the lysogenic life cycle, the genome of the virus will integrate into the host cell's DNA. And when it's integrated into the host cell DNA, we call it a prophage. That's when a lysogenic virus, its DNA, is inside the host cell's chromosome. We call it a prophage. And now, whenever the cell replicates, the prophage DNA also replicates. And this is the only time that a lysogenic virus replicates. And that's when the host cell replicates. So that each daughter cell will receive a copy of the lysogenic virus's DNA. Any question about any of that? Now, for reasons that are not well understood, every once in a while, the lysogenic genome, or the lysogenic DNA, will come out of the host cell's DNA, as shown here. And then the lysogenic virus leaves the lysogenic life cycle and enters the lytic life cycle, where the virus actively replicates, lyses the cell. Any question about any of that? As I stated, it is not well understood why the lysogenic life cycle virus leaves the lysogenic life cycle and enters the lytic life cycle. And they've been studying this for more than 70 years, and they've never gotten a real good handle on it. So let's go into that in a little more detail. Oh, where did I begin? Here we go. The lysogenic virus has injected its host cell, the viral genome into the cell. We're going to talk about it as viral DNA just because it makes it more easier to discuss. The viral DNA integrates into the host cell's DNA. When that happens, we call it a prophage DNA. The lysogenic viral DNA is inside the host DNA. Okay? And then the only time that prophage replicates is when the host cell replicates. So one cell replicates its DNA and then replicates into two daughter cells. Each daughter cell will get a copy of the prophage. And this can go on through many cell divisions. Eventually, though, for reasons that are not well understood, the prophage gets excised from the bacterial chromosome, and then that initiates the lytic life cycle. So the cell no longer is in the lysogenic life cycle, it enters the lytic life cycle. Let me state that there is a gene of the lysogenic virus, which is a repressor protein, and it shuts down the genes of the lytic life cycle of this virus. And if ever that repressor protein gets interfered with or disrupted, that is when the virus leaves the lysogenic life cycle and enters the lytic life cycle. Any questions about that? There, that's stated right there. The repressor proteins keep the lytic life cycle genes dormant. All right, let's watch the rest of that video talking about the lysogenic life cycle.
Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any questions? There are a couple of consequences of lysogeny. First, the lysogenic cell, we're talking about the bacteria cell, is immune to infection by the same bacteriophage. So once the cell becomes a lysogenic cell, including the prophage of the virus within its DNA, this cell is immune to infection by the same lysogenic virus. And then phage conversion can happen. The host cell, the bacteria cell, has picked up a new gene or new genes from the lysogenic prophage. And that can give this cell new properties, and we call this phage conversion. An example of this is the production of a phage-encoded toxin by the host cell. And when Corynebacterium diphtherium produces the diphtherium toxin, it's because the diphtherium toxin is encoded by a lysogenic virus. So the only way that Corynebacterium diphtherium can produce the diphtherium toxin is if the bacteria is infected by a lysogenic virus. Any question about that? Corynebacterium diphtherium, if it doesn't have the lysogenic virus, cannot produce the diphtherium toxin. And that is a consequence of lysogeny. Are there any questions? All right. If there's no questions, I'm going to end it here, and I will be in the lab from 6:30 to 6:45 to answer questions. If there are no questions, you do not need to come to the lab. And if there are questions, I will be there until the questions are ended. But if there are no questions, I'll log off at 6:45. All right. I will see you.